Hi, Eliza. How are you? Sending link. Good to hear. Okay. Just gonna wait a little bit. Have everybody trickle in. Oh, I'm good. Thank you. So I just wanted to give you guys a little bit of a heads up. Um, so Lily, I know at least for you tomorrow, there will be a little bit of a tricky situation with your guys' microgens, your little microgen tests. So what wound up happening was um somebody in the lab, I don't know exactly who, but they left the microgens in the incubator for too long. And what happened was the wells, they started to dry up. So when the wells dried up, it can skew your results. And what's supposed to happen is that there's at least a little bit of liquid left, and you're supposed to add reagents to it and read the results, right? But because of it being dried up, we're just going to do the best we can. We're going to continue as if everything was normal, and you guys won't be um, reprimanded, or you won't be told that what you did was wrong, because it wasn't your fault that your results are going to be a little skewed, and you might have a little bit of a headache. Pack some Advil. It's going to be a little bit of a doozy tomorrow. Um, but I did want to preface yeah, you guys will get a chance to do it again, and hopefully that time that they don't dry up. But cross our fingers, knock on wood, right? Just to preface what the lab is going to look like tomorrow, you'll come in and you will first do your antimicrobial lab. So that's going to be lab 13, I want to say lab 13 or 12, where you guys are going to do the similar technique with making a lawn out of your bacteria with the McFarland standard. You guys will do that yet again. So McFarland standard and skewed results definitely bring some Advil. But you'll make your lawns. The one thing that I definitely saw with results for the antimicrobials, the one thing that I saw was that the lawns didn't turn out as thick as we'd hoped. And sometimes they were very, like, we almost didn't see any bacteria growth. And it's not that the antimicrobials were so strong that it killed all the bacteria on the plate. The point of this lab is to show you that things are gross, and that what we have to kill them may not be might not work too well. But that just depends on what you use, right? So that's the point of this lab. So there's no way that it could knock out the entire plate. So you guys will start with that, reading and interpreting that, and then you guys will move on to your, I'm sorry, you guys will do your antimicrobial first. And what I'm trying to say is that learn from your antimicrobial lab and make sure that you paint a really big, thick lawn. Remember, you're gonna go, here, I'll draw it up. So you'll go one, right, all the way across. Make sure you fill it in nice, no gaps. This has gaps because I'm not trying to run this pen out. You're gonna turn it, right? And then you're gonna go here, all the way across, nice and thick. What you really want to do with your lines is that you want them to be like so close to each other you can't see through them. Like even going back on yourself is okay. Like you just don't want anything through. But remember that you're not done after just two. You're gonna hit this little 45 degree, and go like that across, so that we're covering this up. Yeah, so sometimes it will feel like it's just skipping. Sometimes what you have to do is, say this is my plate, right? This is my plate. What'll happen is if you're pushing too much of an angle, it will skip. The friction will cause it to like kind of skip. And then what you want to do to kind of try to correct that is kind of like make this try to go as like parallel as possible, and try to just let the, not necessarily push down, but just let the weight of the swab itself touch. Yeah, you got it. And then I had the question, is it possible we have to make a lawn on the lab exam? So for this lab exam, I don't believe so. She's also not going to make you do a McFarland standard, but that could change. But I know for sure no McFarland standard. I don't know for sure about the lawn, but I'm willing to bet no, because it's one of those things. It's we initially gave you plates to streak and isolate because we wanted to see your technique. We wanted to see how you were picking up lab technique, right? But with this one, it's more of just like, it just depends sometimes. So I don't think so. So that was the one thing that I wanted to reiterate, because it seemed to be a common theme. If I see you guys again, I'll try to get Dr. B to show this picture, but I have pictures of what my lawns used to look like, and it should be very thick. You shouldn't have any, like if you were to look at the plate, a thin lawn, you would be able to see where your swab went and what you missed. Does that make sense? So like if you were to like look at the edge of your plate, if you were to look at the edge of your plate, let's say that's like the edge, right? And this is where your swab went. And then let's say you came back and you did this, right? And you don't see any growth here, but this is all growth, right? Oops, sorry. So this is like really zoomed in on that edge of the plate. Let's say you went your swab this way, came back this way, right? You didn't hit this little corner right here, and that's a no-no for lawns. We want to see it packed full of bacteria. Okay. So that was my little 411 on that one. So let's see. So yes, your antimicrobials. After the antimicrobials, you guys took a look at the the antibiotics lab. That's what you're painting a new lawn for. The antimicrobial lab is the one that you guys read after you do your next lawn. And when you guys read that, don't be surprised that you have some weird, funky looking results. It's not going to look like a circle. It's not going to look like the lab PowerPoint. And if it does, great. That means that you're doing what you are supposed to be doing. Keep doing it. But it's okay if it doesn't look like that. What you really want to try to do when you are measuring it, because you have to measure in millimeters, the zone of inhibition. So for that zone, the two key differences that you will go over will be if it's bactericidal versus static. So bactericidal, there's going to be a clear defined line and there's going to be no growth inside of that line. It's going to be very black and white. For bacteriostatic, you could see the formation of a line, but it doesn't necessarily mean that there's no growth inside. You might see a colony or two. You might see that parts of the circle are a little more indented than others. And that's completely normal. That just means it's bactericidal, or sorry, static. Bacteriostatic means that it's not black and white. So an example of something that's bacteriostatic would be our refrigerators, right? It keeps our food at a decent temperature so it doesn't grow, right? If there was any bacteria already there, then that means that it's still there. It's probably still alive, but it doesn't grow. So that's another thing with the zones. but you might wind up with like a funky looking zone. Like here's your filter paper, and then this is your bacteria, and then you have like this weird gap, like 1 or 2 in here, and then like another filter paper here, and then like this is completely swamped with bacteria around it, right? So the best way to measure this is to see what the least temperature is. Because what could have happened was your filter paper was overloaded and it kind of spread all over the plate. That's very possible. So what you would want to do is you would want to take your ruler right here, measure from the middle of the filter paper to the edge of the, like, kind of like the, not necessarily the closest bacteria, but the closest line, right? So this is, let's say like that was kind of how it looked. And then it like spread out here. But let's say there's one right here. You wouldn't measure to like the closest bacteria, but you see how there's kind of like a circle right here. It looked like the antimicrobial diffused out a little bit. That's what we want to see. The kind of diffusion ring, right? It didn't diffuse out right here correctly, whatever might have happened, but right there it did. So you want to measure here to here, but that's just the radius, right? Remember math. That's just the radius. We want the diameter. So you'll take that number and multiply by 2, and that's what you'll put on your reports. Okay. So that's that. Does that make sense? It's going to make a little bit more sense, I think, when you get into lab. I also understand that that's like weird. My computer keeps like going back a little. But yeah. So in all, that would be your reading of the antimicrobial lab. And then after your antimicrobial lab, you'll go to your TSIs and your microgen tests. So I already told you guys about your baby microgens that might have died. Not all of them though. We're gonna still continue on. We're going to, the show must go on, right? We're gonna add the reagents correctly. If you guys want to write this down, it'll help you. So I want you guys to remember that Kovacs is one of your reagents. Kovacs is going to be 60 seconds. Okay? You're going to have a VPA 1 and 2. That's your step 1 and 2. Don't quote me if it's VPA, because I think there's another one that starts with a V that I can't quite remember. But VPA 1 and 2, your A and B, they're the same. 1 is A, 2 is B. That's going to be your 30-minute one. That's why we like try to rush you on that one. And then your last one is going to be your ferric chloride. That's the FeCl2. It goes by another name. Don't quote me on it, dang. But it goes by another name. It should be written on the board, and I tried to underline it and like star the right things so that you guys can see it. But just double check it on the board. Just remember those times. Those times is how long you wait to read the result. What kind of happened today was that some people didn't read the results on time, so they got the wrong result. So just make sure that you set those timers. So 60 seconds for Kovacs. Kovacs smells. I hate Kovacs. Then your step 1, step 2, step A, step B. That one's going to be 30 minutes. And then your last one is going to be that ferric chloride. And that's just 60 seconds for that one. Okay. So that one's a good one to remember. So when you bring it to your table, 4 reagents, okay? Those reagents are added and they give you a response, and that goes on your little chart. You'll learn how to write on the chart. It's pretty simple in class, but that's kind of the outline of what you'll be seeing tomorrow, what you saw today. but the main point of today's Zoom session was to cover the P-Glow lab. But before I start that, is there any other questions that you had from either today's lab, you have going into tomorrow's lab, anything from the homework, any from the labs? Reminder that the P-Glow lab that we're going to go over is due tonight, I believe. Okay, it's due tonight for Monday, Wednesday, due tomorrow for Tuesday, Thursday. So is there any questions that I have? All good? Okay. I did want to briefly mention the quiz. So you guys, so Tuesday, Thursday, we'll go over the quiz. Tomorrow, Monday already did. Okay. But for the quiz, I did notice that, you know, I did hone in on certain things during the review sessions, and if you were there, I hope you got those questions. But the one thing that I didn't go necessarily in depth on was the sugar backbone, what is on the 5' and what is on the 3'. So the 5', I think because she stressed it a lot, and I asked her how many people had missed the question. She said not that many, but she said it was very important to know. The 5' end, that end has the phosphate group, and the 3' has the hydroxyl group, and that's specific to DNA. But yeah, DNA, just remember those two. Okay, so let's go ahead and hop over to the transformation lab. Okay, so what type of operon did we mention was the this type of operon? We have, yes, good job, inducible. I was about to say we have two different kinds. So yes, this lab dealt with an inducible operon. So just to outline what kind of we did, we took this jellyfish, right? This poor innocent jellyfish. I'll give him spots like in SpongeBob. Okay, so we took this poor innocent jellyfish and we decided to take a gene out of him, right? So we got this gene. We took this gene out of him, right? And that's going to be our GFP, our green fluorescence protein gene. Fluorescence protein gene. Okay. So we isolated this from the jellyfish and we put it into a plasmid. So we stuck this bad boy into this plasmid. Right? Remember, plasmids are circular. They are smaller and they just are DNA, right? They're kind of like the nucleoid or the chromosome of a bacteria. It's just smaller and it contains less DNA. So we put it into a plasmid. Now we're going to label this P-Glow. Okay. So now what we did was we took this plasmid, right? Here's our little E. coli. It has our paratrachus flagella. Remember that one? Right? Here's its normal circular chromosome. Right? So we took this P-Glow plasmid and we stuck it right inside. So now, how did we stick it inside, right? So this is how we had that artificially created from a bio wall, right? Now this E. coli needs to be, what do we call it? What do we say that this E. coli or any bacteria has to be in order to accept a any type of bacteria?
Competent.
There you go. So the E. coli, so competent E. coli. competent E. coli can now receive this pico plasmid. Oops, pico. There you go. So now we have this plasmid in here, right? Now, how did it get in? So we made it competent with our CaCl2, or did we? Yeah, we did. So we made it competent with our CaCl2, and let me look at the procedure real quick, just so because there's a couple of the ones that I confuse sometimes. Okay, so sorry, I'm pressing. I don't want to tell you guys something wrong. Let's see. So yes, it's CaCl2. Okay. So the CaCl2, right? We put the E. coli in the CaCl2. Okay. And that is that allows it to become a competent cell so it can go inside, right? All right. So now, remember we had to keep this guy on ice, right? So I'm just gonna draw little ice cubes. Okay. Little ice cubes. Pretend those are shiny ones. All right, ice cubes. We had to keep this guy on ice. So we kept it on ice cold CaCl2. And now what we did to this bacteria was we heat shocked it. So we took it out of this ice and stuck it in a nice little bath, right? Nice little fancy bath. Like I don't know if you guys like them, but they're like claw tooth bathtubs. Like the claw foot, that is. So there's our E. coli sitting in a little bathtub. And it's got a book on the side. Who knows? A book relaxing. Not really relaxing. He doesn't like this at all. He hates baths. Oh no, it's too high for you to see. Okay. Our E. coli's in our heat shock bath. Okay. So we heat shock this guy, and his membrane gets all frazzled, right? It opens up like your pores. So when we heat shock him and then put him back into his ice, sucker, when we heat shock him and put him back in the ice, he's not gonna like it, and all of a sudden his membrane's back closed tight, right? So that allows this plasmid to be fully taken up and incorporated. Okay, does that make sense? Does our procedure make sense? And then from this water, from the I remember from this point we plated it, right? Okay, cool. So yeah, that's our little visual on how we did this, right? And remember that the E. coli was mixed with, what was it mixed with? It was like, does anybody remember what it was mixed with? LB broth. That's it. That's what I was missing. The LB broth was what we mixed it with. Okay. So now let's take a look. I'm gonna kind of like copy this over. So this makes sense. Everybody's got this. I'm gonna go ahead and erase it. I know my little drawing in my back is gonna be gone. Okay. So now we have our 4 plates, right? I'm try to see what's the edge. Okay. Oh no, I'm gonna run out of room. You guys see it? Yes, you can see that one. I'm just gonna leave it with that. Okay. So we plated our 4 items, right? So this was our LB amp, and then we had another LB amp. Now I'm gonna make that underneath here. So this would be our other LB amp. And then we had the LB amp era, right? So part of that pico gene was a beta lactamase enzyme, or not enzyme, but how to make it, right? So essentially in that plasmid, we taught E. coli how to make a beta lactamase, right? So the one thing you guys will learn is that in ampicillin, which is a type of penicillin, they all have a similar structure called the beta lactamase ring. Now, what happens if you have beta lactamase? It cleaves that ring. So when it cleaves that ring, it renders the antibacterial useless. It can no longer impact the bacteria at all. So keep that in mind for the ampicillin. This plate has ampicillin, but the organism itself has the ampicillin resistance gene, which is it creates beta lactamase and cleaves that beta lactam ring. Okay, keep that in mind. And our era, what was that? What was our ARA? The arabinose. What was that in this plate? Yes, it was the sugar. It is the sugar. Okay, so we have the arabinose sugar, we have the ampicillin in this plate, and we have our just nutrient right in this plate. In these 2 plates, we have the nutrient and the antibacterial. This plate is just nutrient, right? So if we were to plate right, this is P glow negative. We'll say this is P glow negative. This is P glow positive. And this is also P glow positive. So knowing normal E. coli, right? It is not normally bioluminescent, right? So do you think it would, if it's P glow negative, do you think it would grow on any of these plates? Even if we had plated it over here, do you think it would glow at all? GLO glow. Exactly. So it physically cannot glow under its normal circumstances. So there should be no glowing between these 2. Okay. Now growing, on the other hand, right? We know that E. coli is sensitive to penicillin. Okay. So if we know that that E. coli is sensitive to penicillins, and especially ones with a beta lactam ring, what's going to happen if we put it into our plate? We put E. coli plus our ampicillin into a nutrient plate. But E. coli doesn't like ampicillin. Ampicillin will kill E. coli. What would happen to this plate? Would we see anything? Exactly. So no, we wouldn't see anything grow. So this plate is going to be absolutely empty. However, this is just a nice little nutrient broth. I normally coalesce this one. So we'll see some growth, right? So we will see some growth on here, on our just normal plate. No growth here though, because our ampicillin killed an E. coli that we plated that was not transformed. On the other hand, we transformed the bacteria, right? Our positive P glow. This bacteria got the plasmid that contained the DNA structure in order for it to learn how to code for beta lactamase, right? So it can cleave that beta lactam ring. All of a sudden, it's invincible. You can't stop it. So what do you think is going to happen if it has that beta lactamase to this ampicillin?
It's going to grow.
Yes. So it'll render this ampicillin useless and it will grow. Okay. So that's one of our key differences here is that this showcases our ampicillin resistance gene. So this is going to showcase our AMP-R, which is not just ampicillin resistance, right? It's just our, here, I wrote that too low. It's not our ampicillin resistance. It's the coding for beta lactamase, which cleaves any beta lactam ring. That could be penicillin, that could be methicillin, that could be, what other penicillins are there? You get the gist. Anything with a beta lactam ring, right? I'm gonna put that to where you can see everything. Okay. Now here, what special little thing did we see over here? We obviously saw growth, right? But we also saw glowing. So all of a sudden, I'm just gonna put like a little gradation, right? We saw growth and glow. But what made this one different from this one? Yes, the sugar, right? The arabinose sugar. That is what made these two plates different. So now, if we go to look at you guys will look at my screen. Let's go look at this, right? This doesn't really do it justice, and I'll probably recreate it here in a second. There's no other. Okay. Well, I'll recreate this for you guys, but these should have been your expected results here. Now, unfortunately, Zoom hates me always. So I'm gonna give you guys a quick 5 minutes breaks, and then I will lead off with the next half hour. Well, it'll be more like 25 minutes, but with the next 25 minutes, I will go through the inducible operons specific to the GFP, okay? And the peak glow. So you guys go ahead and take 5 minutes, relax, come back. 5:40. 5:40 is good. Okay. I will be back in 5 to 6, okay?